Hello, everyone. First of all, thank you for attending Novagene's webinar. We are all focused on third generation sequencing, TGS for short, and it's a guide to whole genome re-sequencing. So let's get started with a quick introduction of whole genome re-sequencing. Whole genome re-sequencing offers a full spectrum of genomics variants of organisms, including human, plant, animal, and microbe, through the alignment of a whole genome's readout to the non-genomic sequences, so-called reference genome. Due to the availability of the reference genomes, the whole genome re-sequencing has been applied in study genetic disease, population evolution, and so on, by nailing down variant calling and annotation. Technically, both next generation and third generations are eligible to generate arrays for re-sequencing bioinformatics, while there are disparities linking to the research targets deserves careful considerations. I'm going to cover this comparison in the first section, specifying what kind of studies TGS can help, how it is applied in the research in comparison to next generation sequencing. Second, I'll go through the re-sequencing service workflow and TGS specifications. Great to know if you start a re-sequencing project. Next, we will learn about Novagene powered real case study using such service, and then wrap it up with Q&A section. Genetics variations differs from person to person, serving as contributors to both health and disease. It happens sporadically at both small and large scale. Typically, for small variants, we have single nucleotide variant deletions and insertion, and they could sometimes be escalated to large variants. So SNVs and indels are located in the regions difficult for mapping, genome mappings, but play an important role in our understanding on evolution, adaptation, and disease. Studying this variation has been dominated by next generation sequencing, NGS technology for decades, driven by its superiority in producing highly accurate short sequencing rates on a massive scale with cost-effectiveness. Yet a suboptimal application in many instances inevitably reveals two major drawbacks: GC bias coverage and limited rate length to span highly complex and repetitive area. A greater sequencing technology is needed for this tricky small variant and large variant detection events. As for large variants or so-called structural variants, we have seen tenants duplication, intersphere duplication, inversion, translocation, and copy number variants. TGS is not only being able to capture all variants type ranging from single nucleotide variant to large structural variants, but also can more sensitively detect variants that are often missed by short rays, like long indel and SVs. And PacBio and Nanopore are two TGS commercial dominants, and we are equipped with both of their sequencers. In comparison between next generation technology, Illumina sequencer as major representative and TGS, to dedicate Pondix, PacBio, and Nanopore, the most marked characteristics of TGS is a long read length, at least 15 kb, compared to the paired-end 150 BP or 250 BP fragments of NovaSeq. About variant calling, we just learned a little bit in the last page. Illumina is able to accurately detect SNV and indel, but lower accuracy on the complicated SV. While TGS is good at the coverage of a highly repetitive and complex region. Without PCR amplification involving TGS, no GC bias and amplification bias provide more terrific uniform coverage and a high contiguity. Theoretically, TGS is also flexible for cycle efficiency and simultaneously epigenetics characterization. Since TGS is able to detect native DNA fragments and reflect diverse DNA methylation types, these epigenetics signs can be marked as sequencing bases in real time without any specialized library preparation step like basophil conversion. As I mentioned PacBio and Nanopore many times so far, I like to go further specify the extinguish filtered sequencer we currently use, elaborating how to choose the optimal one for your product. Apart from some advantages of TGS technology listed in the previous table, it is worth noting one of the PacBio statistics more than 99.999% Q50 and single molecule resolution. How does it achieve it? The smart cells are partnered with millions of teeny vials called zero-mode waveguides, ZMW, that allowed individual molecules to be isolated. Laser lights travel through the bottom surface of the ZMW, but not completely penetrate it, since the ZMW dimensions are smaller than the wavelength of light. Therefore, it allowed selective excitation and identification of lights emitted from nucleotides recruited for the long elongation. These figures are more straightforward. The library structures like the top figure and the binding of the DNA polymerase and the template DNA strain is anchored to the bottom glass surface of the ZMW. Each of the 4 nucleotides are labeled with different fluorescence dye so that they have a distinct emission spectrums. After adapters bind to the polymerase, fluorescence labeled nucleotide associated with the templates come into the active site of the polymerase. A light pulse is produced to identify the base, and the fluorescence output of a color corresponding to the incorporated bases is evaluated. With translocating to the next position, the next nucleotide associated with the templates initiating the next cycle. And next to the nanopore, nanopore sequencing is a unique scalable technology that enables direct real-time analysis of long DNA and RNA fragments, generating ultra-long reads and real-time data. It works by monitoring the changes to the electrical current as unzipped DNA string molecule is passing through the protein nanopore one base at a time. The resulting signal is decoding to determine the type of bases. The sequencing mechanisms enable the long length reads. It forms in the narrow pore with perfect processivity, and narrowed order of the polynucleotide is reflected in the detected signal. All right, come to the next sections. I'll share how to start your re-sequencing project on TGS platforms. Innovagene will provide a one-stop re-sequencing solution on TGS platform from sample submission to data analysis. Here, the workflows allowing to learn more about how sample is converted to desirable data step by step. First, we accept a variety of sample types you sent to us, and we have a sample submission guidelines posted on our web that outlines best practice and requires indicators for each acceptable sample requirement, sample types. Upon receiving your samples, we conduct sample quality control for library preparation using compatible keys for the sequencer you choose. In terms of the sequencing, there are 2 PacBio sequencing modes, particularly to different variant calling that will be presented later. Then is data quality QC, data quality control, conducted before data releasing. And then it's about our bioinformatics analysis. We offer standard variant calling analysis and custom bioinformatics analysis. This is a demo of sample submission guidelines listing the minimums, amount, volume, and concentration, etc. These are relatively higher requirements than the sample of NGS. And our technical support scientists will also double check the details with you when sample being scanning. In sample QC method, we use QB to determine concentration, gel electrophoresis for purity and integrity, field gel electrophoresis for DNA size determination, and nanopore for sample purity. Just to give a small example here, the table in QC report like this, and it outlines the general information and the result summary, where the sample could be judged as pass or fail, followed by the explanation of library quality. We give several binary scenarios commonly presented on the gel electrophoresis result figures. If you are confused by some of the situation or experienced, the PC teams are glad to figure it out for you. In this page, we concluded specific indicators in 4 categories: total amount, concentration, purity, and integrity, and their corresponding risk for file samples. We'd also suggest the potential solutions for reference to these conditions. But I'm not going to break it down further. You can check and use this table if you unfortunately had a file sample. With PacBio whole genome sequencing, we prepare the library starting with uniformly sharing the high molecule DNA without the amplification step. The fragments then are treated with DNA damage repair and repair adapter ligation and purification. An optimal size selection steps allows shorter fragments to be removed before sequencing in order to favor data output. The library is double-strand DNA templates capped by hairpin loops at both ends. The library is structural linear, but topologically circular. So the hairpin adapter that are ligated to each DNA fragment provide common primer binding sites for DNA polymerase to initiate sequencing. The first sequencing mode of PacBio is to generate reads as long as possible, whereby the individual DNA fragments are only read once, so-called continuous long reads, CLR for short. This method is particularly suitable to detect structural variants, and the accuracy is about 85%, while the error happens randomly, could be corrected by increasing sequencing depth. Alternatively, it can also be proceeded to circular consensus sequencing, abbreviated CCS. At another extreme, for the smaller insert size, the library allows multiple reads to be generated on each molecule. These multiple passes may carry errors in particular size, but they can be combined and corrected by each other to generate a consensus rates for the templates, defined as the high fidelity rates, high-fi rates, with fabulous single molecule accuracy and the least GC content bias, as well as the sequencing complexity bias. In nanopore sequencing strategy, the libraries are prepared in a more consensus way. High molecule weight DNA is optimally fragmented and selected by size, after which end preparation and nick repair proceed to the library. With ligation addition, the library is outside for sequencing. Right before sequencing, the libraries was checked with bioanalyzers for size distribution detection and the qPCR for quantification. The qualified libraries will be proceeded to sequencing. To sum up in this table, you can choose a PacBio or Nanopore by aligning their specification to your needs. Look up the ray length and the accuracy. If you're expecting longer rays, Nanopore won't let you down. And high-five rays from PacBio CCS mode perform well on SNPs detection and indel detection that Nanopore has nothing to compare to. However, they both have shortcomings that you should be aware of. The throughput per cells of PacBio fluctuated partially by species and sample quality and has a little higher price. Nanopore rays are unable to define the indels detection and unable to have a perfect coverage in the low complexity run. Based on our experience, you could decide sequencing depth by variant calling you want or the research target. For example, if you want to detect SVs, you can either use at least 50x sequencing depth on PacBio or 20x Nanopore. Or if you focus on tumoral genesis, the more than 20 times sequencing depth is highly recommended. This is a data quality control, and data quality control reports will be delivered regardless the projects with bioinformatics or not. The two tables summarize the basic statistics of CCS rays and high-five rays. The three visuals help to clarify the ray length and the distribution. In standard analysis, we offer reference genomes mapping and SNPs index, indel index, SV index, CNV index, SN detection, indel detection, SV detection, CNV detection, and corresponding annotations. The high-five rays, CLR rays, and nanopore rays are theoretically able to detect all the variants, but they do have their preference according to the design purpose and the practical performance. High-five rays are doing well on SNP and indels SV detection, while the CLR rays are popularity used in SV detection. Nanopore rays can recognize SV and CNV detection well. In mapping analysis, the sequence arrays align to the known reference genome, and the mapping statistics are manifested in the table of the data report, followed by detailed notes for each definition. In germline mutation detection, the number of SNPs, indel, SV in various genomics regions are calculated and presented in the table, followed by same-day detailed notes, and also followed by the visualization. Following genomics variant detection, annotation of variants in multiple aspects are performed, including visualization of quality or length distribution, mutation frequency of each type of variants, as well as the comprehensive visualization at the genomes level. For BS software used for TGS resequencing, you could check this table later. Next is your case study sharing. The paper I'm going to dissect was published in 2020. Note 2, NLC, GCC repeat expansion are associated with sporadic essential tremor, variable disease expressivity on long-term follow-up. The study screened 1,662 subjects and identify pathogenics NOC2, NLC, GGC repeat expansion with nanopore sequencing and a variation analysis. The TGS technology is optimal for this study, since the large structural variations such as copy number variations, copy number neutron inversions, mobile elements insertions, deletion, translocations, and combination of any of these events are more challenging to detect and characterize compared to the SNPs. So nanopore with long incurrence rates is adept at identifying large and complex SVs with high sensitivity and specificity. This study focuses on the essential tremor, ET for short, that remains a clinical diagnosis for patients who typically exhibit no other neurological signs apart from tremor, while more potentially develops a neurological disorder in the future. ED is considered to have a strong genetics component, as more than half of cases report a positive family history. However, identification of pathogenic variants in ET has been challenging due to its clinical and genetics heterogeneity, variable expressivity, and a lack of pathological hallmarks. Besides the finding on genetic lossy and genes associated with sporadic and familiar ET are either rare or not constantly replicated. And recent studies has reported GGC repeat expansions in the 5 regions of human-specific NOTE2 NLC gene to consegregate with disease in ET families or familial ET pedigrees. In particular, the association of NOT2 NLC with sporadic ET cases has not been investigated, and it is uncertain if the repeat expansion can be identified in the clinical well-characterized ET pedigrees. Therefore, the research into the association of NOT2 NLC with sporadic ET cases and the clinical characterization of the repeat expansions. And all the ET cases were screened for NOT2 NLC GGC repeats using repeat prime PCR, after which cases with intermediate and positive expansions were sent for confirmation using long-range sequencing. This is a nanopore sequencing capable of detecting complexity SVs with a high sensitivity and specificity in the difficult regions, which is a severe limitation of NGS due to the short sequencing length. Then in the studies, they carry out the primer fluorescence arms PCR applicants length analysis. Characterization of SVs is crucial to these studies of the neurological disease. However, due to the short sequencing length, NGS pose limitations on the study of these complex SVs, particularly those involving repetitive regions. So the studies greatly depends on the TGS. Out of over 1,600 subjects and 200 healthy controls, they identified the GGC repeat expansion of more than 80 units in 4 sporadic ET patients. Notably, Patient 4 was carrying 107 GGC repeats shown in the figure, and the tremors remained stable for approximately 10 years before progressing in association with mild cognitive decline and motor slowing. All of the 4 had mild to moderate hand tremor with no systemic involvement. It's a reminder possible that the age at the tremor onset and the repeat size may partially account for the variance. And in conclusion, the study highlights the observations. Note 2, NLC GGC repeat expansion can associate with a sporadic ET, while remains rare in the clinical well-characterized ET categories. The third-generation sequencing technology have the capability to produce a substantial longer reads. Such an advantage has a critical implication for both genome science and study of biological in general. Today's webinar just pinpoint to one of the TGS services we're confident to provide, whole genome sequencing on TGS. To add this, we also have TGS pipeline for RNA sequencing, targeted sequencing, complex populations, and epigenetics services. So feel free to contact us if you're interested. Before QA section, I'd like to present our capabilities that you may concern. We are dedicated in sequencing service for more than 10 years with cutting-edge genomics technology and high-performance computing cluster to improve life science research and human healthcare. We have several major labs all over the world and have worked with almost all top 100 scientific research institutes and top 10 global pharmaceutical companies, and helped over 950 SAI articles. Here's a link on our web to the papers. To ensure efficient and stable delivery, our Falcon fully automated systems works so hard, 24 hours a day and 7 days a week. Another contributors is our world-class data center for your fast, accurate, and secure data delivery. All right, last, I'm going to wrap it up by addressing some of the frequently asked questions. And I see the questions in a QA module. Like, what mapping rates would allow the variance calling? Could you please list the potential cause of a low mapping rates? We highly recommend the mapping rates ranging from 70% to 90% for variance identification based on our experience. Technically, also, as long as the sequence arrays can be aligned to the reference genome, it would enable the variance calling even under the situation the sequencing arrays are mapped to the reference genomes of another species in the same genus due to the lack of the annotated genomes. So just a higher mapping rate will allow to detect more variants, indicating more of them could be used for the subsequent advanced analysis. And for the potential cause of a low mapping rate, it could be related with poorly assembled reference genomes or the relatively far genetics relationships between the reference and the sample. And alternatively, the treatments to the DNA may change the sequence, like basophil treatment, and herein affecting the mapping rates. Or the sample are contaminated with another species for example, the contaminations of bacteria in plants. And I see here's another question. What if my sample can't meet your requirements, especially as I mentioned, the high sample requirements is that for TGS platform? It's a great question. Per our sample submission guide, the past samples would allow us to ensure the success of a library preparation. However, the hold and file QC results doesn't imply the library preparation or the sequencing has to be hosted. For example, if the samples are insufficient amount of the library preparation, we actually can offer the low input or ultra low input for the library preparation. And to deal with the impurity in DNA sample, magnetic beads or the columns chromatography are available for purification. And our product coordinators will inform the potential risks in advance and offer the constructive suggestions in terms of the QC results and the previous sequencing experience for you. So if you have any extraction issues, you can also consult us for advice. We are always willing to support our clients with our strong scientific expertise. So please don't worry about it. And okay, I will wrap it up with the last questions. Can 4 methyl-cytosines and 5 methyl-cytosine be detected with high-fat rays directly? Any bioinformatics analysis you provide for this? The answer is yes. Since it uses a deep learning model that integrates the sequencing kinetics and incorporates base context, the PacBio sequencing simultaneously captures the epigenetics modification in native DNA without special preparation treatment like bisulfite conversions. High-fat rays have access to multiple analysis of methylation tabs powerfully and efficiently. And we do offer DNA methylation analysis, including single sample methylation analysis, i.e. Methylation density, methylation level density, methylation level of genomics features, and methylation level of up or downstreams, etc. And detection and analysis of methylated sites, like correlation and PCA analysis or motifs analysis, etc. And DMR analysis. And as well as the goal and the key enrichment analysis. So hope my answer is helpful for you. All right, so feel free to contact us if you have any questions about TGS or Novogene. And now I'm glad to give time back to Brianna.